Welcome to Sleepy Stories. In this episode, we will take a clear and steady look at the world of bacteria and microbes, the oldest and most abundant forms of life on our planet. The goal is to help you learn while you rest. If you do not fall asleep right away, that is completely fine. In the time you remain awake, you will discover how these microscopic organisms sustain the air you breathe, the soil beneath your feet, and even the health within your own body. Simply listen, let your breathing slow, and allow the information to settle at its own pace. If you find value in these quiet explorations of science, consider subscribing and giving a like. It helps us continue producing calm, carefully researched episodes for you. The story of microbes begins billions of years ago, long before animals, trees, or even algae existed. At that time, the planet was still forming, its crust unstable, and oceans newly born from endless rain. Volcanoes exhaled steam and sulfur. Lightning struck the thick sky again and again. Out of this restless landscape emerged the first microscopic forms of life, tiny units capable of growth and replication. They were the pioneers of biology, setting in motion a chain of existence that has never stopped. Today, those ancient descendants still surround you in numbers too great to imagine. Every breath you take contains airborne bacteria. A pinch of soil hides more organisms than there are people on Earth. On your skin live entire ecosystems that protect you, feed on oils, and quietly compete for space. Inside your digestive tract, hundreds of species cooperate to extract energy from food and to keep harmful intruders away. Although unseen, they are your oldest companions, sharing the same evolutionary journey that began in Earth's primeval seas. Bacteria and microbes are extraordinarily simple in structure, yet their behaviors and chemistry are complex beyond measure. Most consist of only a single cell enclosed by a thin membrane. Within that boundary floats cytoplasm, a watery interior filled with enzymes, proteins, and genetic material. There is no nucleus, no visible organization, yet each cell is a precise machine, able to sense, react, and divide. Some species convert sunlight into energy through photosynthesis. Others survive by consuming iron, sulfur, or methane, turning inorganic minerals into the foundations of entire ecosystems. They thrive in boiling springs, beneath ice caps, and even miles below the ocean floor, where pressure would crush any larger organism. If you could suddenly see the microbial world magnified around you, the familiar would disappear beneath a new reality. Surfaces would shimmer with moving colonies, each one a living city. Rivers and oceans would appear thick with invisible threads connecting chemical cycles. Even the air would seem alive, filled with drifting cells carried by wind currents. The quiet spaces, your room, your pillow, the gentle stillness of night, are never truly empty. They are part of a continuous web of life that began before time was recorded. Microbes are not only survivors, they are builders. They created the oxygen you breathe, formed the soil that feeds plants, and recycle the nutrients that sustain every living creature. Without them, the planet's chemistry would halt in days. Decay would stop, oxygen would vanish, and ecosystems would collapse. Life depends on them the way the human body depends on the heartbeat. Constant, rhythmic, often unnoticed, yet essential. Their quiet endurance holds a lesson about scale and importance. Power does not always belong to the large or visible. Sometimes it resides in what works patiently and unseen. As you breathe gently now, consider that each inhalation carries molecules of oxygen first released by ancient bacteria more than 2 billion years ago. Those same elements travel through your bloodstream, fueling your thoughts at this very moment. Life's smallest agents continue their task, generation after generation, never resting, yet allowing you to rest. The world remains alive at every level, even when it seems still beneath your awareness. Bacteria and microbes keep the balance steady, They remind us that life's strength lies not in noise or motion, but in quiet persistence, the same calm rhythm that accompanies you towards sleep. To understand bacteria and microbes, we must return to a time before them, when Earth itself was a molten experiment still cooling from its birth. More than 4 billion years ago, the planet's surface was covered by volcanic plains, boiling oceans, and a dense atmosphere of carbon dioxide, methane, and ammonia. Lightning storms flashed across the dark sky, and meteorites fell like sparks from a cosmic forge. The young planet was unstable, yet within that instability lay the perfect conditions for chemistry to change into biology. The first step toward life was not sudden but gradual. Simple molecules of carbon, hydrogen, oxygen, and nitrogen, carried by rain into the oceans, began to meet and combine. Energy from lightning, ultraviolet sunlight, and hydrothermal vents on the sea floor powered their reactions. Laboratory experiments in the 20th century confirmed that such conditions could indeed create amino acids, sugars, and lipids, the raw ingredients of cells. Over time, these molecules drifted together into small droplets surrounded by thin films of fat. These droplets, sometimes called protocells, protected their contents from the outside world and created microenvironments where chemical reactions could repeat and grow more complex. Inside those microscopic shelters, molecules began to interact in new ways. Some of them formed strands capable of copying themselves, an ability that transformed ordinary chemistry into something resembling heredity. The most likely candidate was RNA, a molecule that can store information like DNA yet also catalyze reactions like an enzyme. In what scientists call the RNA world, small strands may have floated freely in warm pools, copying themselves with many errors. Most copies broke apart, but a few were stable enough to last. The ones that lasted became templates for the next generation, setting the first rules of natural selection. Eventually, these early replicators joined with lipid membranes and simple metabolic reactions, forming true living cells. They absorbed energy from sunlight, from iron, or from chemical gradients near deep-sea vents. The process was slow, perhaps taking hundreds of millions of years. But once a cell found a way to reproduce reliably, evolution could begin its long, endless branching. From that point onward, life was not a fleeting experiment, but a self-sustaining system that would fill every niche the planet offered. The earliest microbes were likely anaerobic. They lived without oxygen and would have found today's air toxic. They thrived in warm, oxygen-free waters rich in minerals, using chemical energy instead of light. Fossils of microbial mats, known as stromatolites, still preserve their layered structures in rocks more than 3.5 billion years old. Each layer represents a colony that grew upward toward the sun, trapping sediment and slowly building the first biological architecture on Earth. If you could travel back to that ancient ocean and hover above it, you would see no movement at all, only shimmering blue-green water beneath a copper sky. Yet beneath that surface, invisible reactions were changing the fate of the planet. Molecules folded, replicated, and evolved. The laws of chemistry had given rise to the logic of life. It is humbling to realize that every plant, animal, and human alive today descends from those first microscopic ancestors. The spark that began in silent, lifeless seas continues in you, in every heartbeat, every breath, and every cell of your body. The continuity between those ancient microbes and the modern world is unbroken. The atoms of carbon that once drifted in a primordial ocean now form part of your living structure. As you rest, imagine that connection stretching backward through time. The life within you is ancient, patient, and persistent. Just as the early oceans held the first whispers of existence, your body holds the same chemical rhythms now, quiet, steady, and enduring through the long night. To imagine the world in which microbes first appeared, you must picture a planet entirely different from the one we know. Earth, 4 billion years ago, was wrapped in a heavy atmosphere that no creature today could breathe. The air was thick with carbon dioxide, methane, ammonia, and water vapor, tinted red by volcanic dust and iron particles from constant eruptions. The sky rarely showed the sun clearly. It glowed through clouds of ash, and the light that reached the surface was dim and copper-colored. Across that surface stretched oceans of warm, mineral-rich water. Their waves crashed against shores of black basalt, still cooling from the planet's formation. In the depths, volcanic vents spewed jets of hot fluid loaded with hydrogen sulfide, iron, and nickel. Around these openings, chemicals mixed in gradients of temperature and acidity, creating natural laboratories of endless reactions. Scientists now believe that these environments, rather than calm tide pools, may have been the true cradles of life. The constant flow of minerals and heat offered both energy and raw material for the earliest cells to form. There were no continents yet, only fragments of crust slowly thickening. Meteor impacts remained frequent, delivering water and organic compounds from space. Each collision reshaped the atmosphere and oceans, sometimes vaporizing seas entirely before rain returned them. Yet the resilience of chemistry persisted. When the surface cooled, the oceans again condensed, carrying with them the traces of carbon-based molecules that would one day evolve into the first living systems. The absence of oxygen defined that early Earth. Without it, the sky lacked ozone, allowing ultraviolet radiation to strike the surface unfiltered. Life could survive only beneath protective layers of water or rock. Deep-sea microbes may have thrived precisely because the sunlight above was lethal. Iron and sulfur served as the main energy currencies, and entire ecosystems existed without a single photon of light. Even today, the descendants of those ancient chemotrophic microbes continue to live in deep hydrothermal vents, showing that Earth's earliest strategies for survival have never disappeared. Geological evidence supports this vision. Ancient rocks found in Greenland and Western Australia contain isotopic ratios that point to biological activity more than 3.7 billion years ago. Stromatolites, layered mineral structures built by microbial mats, record the presence of organized colonies long before complex organisms evolved. In these fossils, each layer tells a daily story: microbes growing toward light, trapping sediment, dying, and then forming a base for the next generation. The pattern repeats for thousands of years, building limestone towers that still stand as monuments to the earliest engineers of the biosphere. The planet's chemistry was gradually transformed by these minute architects. Gases released by volcanic activity interacted with microbial metabolism, producing new compounds that altered the composition of both air and ocean. Methane-generating archaea contributed to an early greenhouse effect that kept the planet warm despite the faint young sun. Iron-oxidizing bacteria began depositing rust-colored sediments on the ocean floor, hinting at the first traces of oxygenic processes that would later change the world. Standing on that primitive shoreline, if one could have stood at all, you would have seen no plants, no blue sky, and no sign of motion except the boiling of water. Yet every drop was alive with potential. The Earth's crust provided the elements, the ocean mixed them, and the atmosphere sealed them under heat and pressure. Together they formed a continuous cycle of energy and renewal, setting the foundation for biology itself. When viewed through this lens, the planet was not merely a backdrop for life, but an active participant in its creation. The chemistry of rocks, the energy of sunlight, and the circulation of the oceans all conspired to make replication possible. The stage was vast, the players microscopic, but the result was irreversible: a living planet. At some moment in Earth's ancient history, chemistry crossed an invisible boundary and became biology. The building blocks were already present: amino acids, sugars, lipids, and simple nucleotides drifting in mineral-rich water. But to become life, they needed order, a way to organize reactions, to store energy, and to preserve the results from one generation to the next. This step, from random chemistry to self-sustaining cells, remains one of the most profound transformations in the history of the planet. Scientists believe that fatty molecules, naturally produced in the oceans, began to form thin, flexible bubbles. These membranes could enclose water and molecules inside, creating tiny chemical chambers. The boundaries offered protection from the chaotic environment outside, while allowing selective exchange of nutrients and waste. Within these microenvironments, reactions could continue longer and in higher concentration, giving chance a better opportunity to build complexity. The first protocells were fragile, breaking apart easily, but they also reformed just as easily. Each time they reassembled, the arrangement of molecules inside shifted slightly, testing new combinations. Inside these primitive membranes, energy had to flow in a controlled way. Chemical gradients between the inside and outside of the bubble acted like miniature batteries. Ions moved across membranes through narrow channels, releasing bursts of energy that could drive the assembly of larger molecules. This interplay of structure and energy created the earliest metabolism, a simple set of reactions that captured energy and used it to build more of the same system. Meanwhile, some molecules inside these protocells began to copy themselves. Short chains of RNA, capable of both storing information and catalyzing reactions, may have taken the central role. Each replication was imperfect, introducing variation. Over time, protocells containing slightly more efficient RNA strands reproduced faster, passing their advantages forward. The first selective evolution had begun, guided not by intention but by chemistry itself. As these early systems became more stable, the separation between the living and non-living blurred. A protocell that could grow, divide, and pass on its molecular instructions had effectively become a cell. It could respond to its surroundings, exploit energy sources, and repair itself when damaged. Once this level of organization appeared, the possibilities multiplied rapidly. Life was no longer a series of isolated reactions. It had become a continuous, self-replicating network. Fossil evidence suggests that true cellular life may have existed as early as 3.8 billion years ago. These first microbes lacked nuclei and organelles, relying on the simplicity of direct contact between enzymes and genetic material. They floated in the oceans, feeding on chemical energy from minerals or absorbing sunlight through primitive pigments. Some learned to join together, forming thin mats on the sea floor. Within these communities, waste from one organism became food for another, creating small ecosystems that mirrored the cooperative cycles still seen today. The essential principles of biology, compartmentalization, energy exchange, and heredity were all established in this era. Every living thing that followed, from oak trees to humans, inherited these same rules. The cell became the universal unit of life, a self-contained, self-renewing system capable of near infinite variation. In those first oceans, evolution had found its medium. What began as scattered molecules had turned into organized entities able to persist and change. The Earth itself had become a factory of microscopic experiments, each cell a new version of a successful idea. Through uncountable cycles of replication, mutation, and survival, complexity was no longer an accident, it was an inevitability. Once life had established itself in the oceans, it diversified with astonishing speed. Evolution began its endless experiment, branching and branching again, shaping lineages that would one day lead to every organism on Earth. The earliest forms were small and simple, yet their genetic creativity knew no bounds. By exchanging and modifying bits of molecular code, they filled every possible niche: hot, cold, acidic, saline, or dark. What emerged from those ancient waters was not a single species, but a vast network of microbial ancestry that still forms the foundation of life today. When modern scientists map the relationships between living things, they trace them back to three great domains: bacteria, archaea, and eukarya. Each represents a distinct strategy for existence, a different interpretation of what it means to be alive. Bacteria and archaea are both single-celled and lack a nucleus, yet their internal chemistry and genetic organization diverge in fundamental ways. Bacteria possess familiar cell walls made of peptidoglycan, while archaea rely on unique lipids that resist extreme temperatures and chemical stress. For a long time, these two groups were assumed to be variations of the same theme, until microbiologist Carl Woese, in the late 20th century, analyzed the molecular sequences of their ribosomal RNA. His results stunned the scientific world. He discovered that archaea were not simply another kind of bacteria. They represented an entirely separate branch of life, as distinct from bacteria as bacteria are from humans. With that revelation, the tree of life gained a new shape, not as a straight line from simple to complex, but as a deeply rooted network with multiple trunks rising from a common origin. At the base of this tree lies a hypothetical ancestor called Luca, the last universal common ancestor. Luca was not the first living thing, but the point from which all modern life descends. It likely lived around 4 billion years ago, in an environment rich in hydrogen, carbon dioxide, and metals. It had enzymes to manage energy, genes to encode proteins, and the ability to copy itself. Every cell on Earth still carries a trace of Luca's molecular machinery. When your own cells read DNA or make proteins, they are repeating steps that began in that ancient lineage. The bacterial domain, which includes countless familiar species, went on to dominate the biosphere. Some bacteria learned to harness sunlight, inventing photosynthesis. Others evolved ways to process nitrogen or sulfur, cycling nutrients through the planet's systems. The archaea took a different path, thriving in extreme conditions where other life could not survive: boiling springs, salt flats, and deep-sea vents. Their tolerance for heat and acidity provides clues to what early Earth may have been like, and perhaps what life could be on other worlds. From these two microbial empires, a third domain eventually emerged: the eukaryotes. Unlike their ancestors, eukaryotic cells developed internal compartments, including a nucleus to protect DNA. Evidence suggests that this leap occurred through a remarkable act of cooperation. A larger cell engulfed a smaller bacterium and, instead of digesting it, formed a partnership. The smaller cell became the first mitochondrion, a tiny power generator. This event, called endosymbiosis, turned out to be the gateway to complex life, to plants, animals, and humans alike. The microbial tree of life, viewed today, is not a tidy diagram but a web of connections. Genes flow sideways as often as they descend vertically, pass between species through horizontal gene transfer. This fluid exchange blurs the boundaries of ancestry, showing that evolution is not just a story of descent but of sharing. Microbes trade tools, learn from one another, and adapt as a collective whole. Every human, animal, and plant remains part of that vast, ancient network. In every breath, in every cell, the same processes continue: replication, mutation, selection, renewal. The tree of life still grows, invisible and unstoppable, rooted in the smallest forms that first made the Earth a living world. Roughly 2.5 billion years after life began, a transformation occurred that would alter the planet forever. Tiny blue-green cells floating in the ancient oceans learned to harness sunlight in a new way. These were cyanobacteria, and they had discovered photosynthesis, the ability to capture light energy and use it to split water, releasing oxygen as a byproduct. It was a biochemical invention so powerful that it reshaped every future possibility for life on Earth. Before this innovation, the atmosphere contained almost no free oxygen, volcanoes and hydrothermal vents filled the air with carbon dioxide, methane, and sulfur gases. For the microbes that thrived in that world, oxygen was not life-giving, it was toxic. Yet, as cyanobacteria multiplied, they filled shallow seas with mats of green film, each cell working like a solar panel. With every sunrise, they drew in sunlight, water, and carbon dioxide, building sugars for energy and exhaling oxygen into the surrounding water. What began as a local process spread until the planet itself began to change color and chemistry. At first, the oxygen they produced reacted with dissolved iron in the oceans, forming rust-colored minerals that settled to the sea floor. These deposits, still visible today as banded iron formations, record the slow build-up of oxygen through deep time. When the iron was finally saturated, oxygen began to accumulate in the air. The event is known as the Great Oxygenation, a turning point that transformed Earth from a reducing environment into an oxidizing one. For many ancient microbes, this shift was catastrophic. Species that had evolved in oxygen-free conditions faced extinction, but for others, it opened a vast new frontier. Oxygen made possible far more efficient ways of extracting energy from food. Cells that adapted to it gained an evolutionary advantage, paving the way for complex multicellular organisms millions of years later. The waste product of cyanobacteria became the essential fuel for higher life. Geological records show that the Great Oxygenation also triggered the first global glaciations. As methane levels dropped and oxygen reacted with carbon compounds, the greenhouse effect weakened and temperatures fell. Ice spread across continents for the first time. Yet even through these frozen ages, photosynthetic microbes endured, continuing their quiet work beneath layers of ice, never ceasing their slow transformation of air and water. When you take a breath today, the oxygen filling your lungs comes from the same process invented by those ancient bacteria. The chloroplasts inside every leaf are their descendants, still performing the same reaction with elegant precision. Photosynthesis is not just a feature of life, it is one of life's greatest collaborations between sunlight, water, and carbon. In a calm and factual way, this connection extends to you as you breathe now. Each gentle inhalation carries molecules that once drifted in ancient seas, released by organisms smaller than a speck of dust. The rhythm of that exchange, oxygen out, carbon dioxide in, is the planet's oldest dialogue, still continuing quietly within every living being. You may notice your breathing slow, steady, and effortless, just as it has been since life first filled the air. From the moment cyanobacteria began their work, the Earth could never return to what it was. The atmosphere had been permanently rewritten, and the story of life entered a new chapter, one where oxygen would power the engines of evolution, and the smallest cells would give rise to worlds of complexity and thought. After the Great Oxygenation transformed the planet, not every organism adapted to the new chemistry of the air and sea. Many early microbes perished, unable to tolerate oxygen's reactive power. Yet life, even at its smallest scale, has a remarkable ability to endure. Some microbes found refuge in hidden environments where oxygen could not reach, deep underground, beneath ocean sediments, or inside volcanic vents. Over time, these survivors evolved into specialists capable of thriving under the harshest conditions the Earth could offer. They are known today as extremophiles, organisms that challenge the limits of what we consider habitable. Some live at temperatures above boiling, others in water so salty it crystallizes around them. Acidic pools that dissolve metal, radioactive waste, glaciers, and even clouds all serve as homes to microbes that seem to ignore the boundaries of chemistry and physics. Their continued existence shows that life is not fragile, it is flexible, able to rewrite its rules whenever the environment demands it. In the scalding geysers of Yellowstone or the hydrothermal vents along the mid-ocean ridges, thermophiles flourish. These heat-loving bacteria and archaea possess enzymes that remain stable at temperatures exceeding 100 degrees Celsius. Their proteins are designed to fold tightly, resisting the breakdown that would destroy ordinary life. In contrast, psychrophiles, the cold-loving microbes, operate at the opposite extreme, surviving in Antarctic ice and the frozen crust of permafrost. Their membranes remain fluid in the cold, and their enzymes work efficiently at near-freezing temperatures. Other specialists thrive in chemical extremes. Halophiles require salt concentrations that would dehydrate most cells. They use unique pumps to balance internal pressure. Acidophiles and alkaliphiles endure pH levels that would destroy organic molecules, yet their internal systems stay neutral, buffered by molecular defenses. Then there are the radiation-resistant species like Deinococcus radiodurans, nicknamed Conan the bacterium. It can repair its DNA even after exposure to radiation doses thousands of times higher than those fatal to humans. The existence of such organisms forces scientists to redefine the meaning of life's boundaries. The planet once seemed neatly divided between habitable and inhospitable, but extremophiles erased that line. They demonstrate that wherever energy can be harvested and matter can persist, life finds a way to build a foothold. In the deep crust, microbes live more than 3 kilometers below the surface, using hydrogen from water rock reactions as their only fuel. Others cling to the walls of nuclear reactors or inside clouds of sulfuric acid. Each environment that appears sterile eventually reveals its hidden inhabitants, waiting patiently to be discovered. The study of extremophiles does more than expand our understanding of Earth. It also guides the search for life beyond it. The same adaptations that allow microbes to survive boiling water or freezing ice suggest that other worlds, Mars, Europa, or Enceladus, might harbor similar forms of resilience. If life can exist in volcanic vents and under Antarctic ice, it may also exist beneath alien surfaces where sunlight never reaches. These remarkable species remind us that survival is not about strength but adaptability. They teach that life's essential strategy is not resistance, but adjustment. Over billions of years, microbes have faced fire, ice, radiation, and starvation, yet they continue to multiply. Their endurance holds the memory of every planetary change the Earth has known. Extremophiles are, in many ways, the planet's time travelers. They carry within their DNA the record of ancient strategies, unchanged because they still work. As long as energy flows and molecules can interact, life persists, quiet, unseen, and infinitely inventive. The history of Earth's surface is not only written in rocks, but also in the activity of microbes. Long before forests grew or animals burrowed, microscopic organisms were already reshaping the planet's crust. Every grain of soil, every mineral deposit, and every fossilized layer carries silent evidence of bacterial work. Life and geology evolved together, the living constantly sculpting the non-living, and the non-living providing the materials for life to endure. One of the earliest collaborations between biology and stone can be seen in stromatolites, layered structures built by communities of cyanobacteria. Each day, these microorganisms trapped fine sediments while growing upward towards sunlight, forming domes and columns in shallow seas. Over time, minerals hardened around them, preserving the microbial architecture. Ancient stromatolites found in Australia and South Africa are among the oldest records of life on Earth, more than 3 billion years old. They reveal how microbes once dominated entire coastlines, turning light, water, and minerals into the planet's first ecosystems. Microbes not only build but also dissolve rock. Many bacteria and archaea extract energy by oxidizing or reducing metals such as iron, manganese, and sulfur. These reactions are fundamental to Earth's biogeochemical cycles. For example, Iron-oxidizing bacteria convert ferrous iron to ferric iron, creating the reddish layers that color ancient sedimentary rocks. Sulfur-reducing bacteria, on the other hand, break down sulfates, producing hydrogen sulfide, a gas that smells of decay but drives vital chemical exchanges in marine environments. In caves, microbes act as silent masons, decorating the walls with delicate mineral formations. Some species use carbon dioxide and water to precipitate calcium carbonate, building stalactites and stalagmites, molecule by molecule. Others etch away limestone by releasing organic acids, widening chambers over thousands of years. Even deep underground, far below sunlight, microbial colonies persist, feeding on trace minerals and leaving their signatures in crystal growth patterns. Modern mining and environmental science continue to reveal how intertwined life and minerals remain. Certain bacteria are now used in biomining, a technique that extracts metals from ore without the pollution of traditional methods. Species such as Acidithiobacillus ferrooxidans dissolve copper and gold from rock by controlling oxidation reactions. The process is efficient, sustainable, and inspired entirely by what nature has already done for eons. Conversely, bioremediation employs microbes to clean toxic waste, using their metabolic skills to break down pollutants into harmless compounds. The same chemistry that once shaped mountains now heals contaminated soil and water. Microbial influence extends even to the deep ocean floor, where entire ecosystems depend on mineral interaction. At hydrothermal vents, water rich in metals gushes from the crust, mixing with cold seawater. Microbes living there extract energy directly from chemical gradients, forming the base of complex food webs. Tube worms, clams, and crustaceans all rely on symbiotic bacteria that convert inorganic compounds into organic nutrients, a process known as chemosynthesis. These vent communities demonstrate that sunlight is not the only foundation for life. Minerals alone can power entire biospheres. The boundary between rock and life has always been porous. Minerals provide surfaces for reactions, shelter for cells, and catalysts for biochemical change. In return, microbes regulate the composition of oceans and the atmosphere, dissolving and reforming minerals with each metabolic breath. Even the chalk cliffs and marble sculptures that humans shape today are, at their core, ancient microbial remains compressed into stone. When viewed on geological timescales, Earth appears alive in a literal sense, a planet whose crust, air, and water are continuously processed by microscopic workers. The slow cycles of erosion and sedimentation are mirrored by equally patient cycles of bacterial metabolism. Together they maintain the balance that allows the planet to remain habitable. The partnership between microbes and minerals is as old as life itself, an unbroken dialogue between stone and cell that continues, quietly, beneath every step you take. The oceans are the cradle and current heart of microbial life. From their depths to their surface, they contain the greatest variety and abundance of organisms on the planet. Every milliliter of seawater holds millions of living cells, a floating galaxy of bacteria, archaea, viruses, and single-celled plankton. They are invisible to the naked eye, yet they regulate nearly every global process that sustains life, from the cycling of carbon to the production of oxygen. The ocean's surface, glittering under sunlight, is home to photosynthetic microbes that act as microscopic forests. Chief among them are cyanobacteria and tiny eukaryotic algae such as Prochlorococcus and Cynococcus. Despite their size, these organisms perform almost half of the photosynthesis on Earth. They capture sunlight, absorb carbon dioxide, and release oxygen, the very same reaction that ancient cyanobacteria once used to transform the planet. In effect, the sea breathes through them. Beneath this bright layer lies a world of heterotrophic bacteria that feed on organic matter drifting downward from above. Every dead plankton cell, every flake of waste, becomes a source of carbon and nutrients. These microbes break the material into smaller molecules, recycling it for reuse. Without them, oceans would fill with debris, and life would soon stagnate. Instead, the microbial community keeps the water clear, fertile, and balanced. Deeper still, where sunlight no longer penetrates, other microbial strategies prevail. Chemotrophic bacteria derive energy not from light but from chemical reactions. Near hydrothermal vents, they oxidize hydrogen sulfide and methane, producing organic compounds that sustain entire ecosystems in perpetual darkness. These deep-sea colonies prove that life needs only energy and structure, not necessarily sunlight, to flourish. Microbes also shape the chemistry of the ocean itself. Nitrogen-fixing bacteria convert atmospheric nitrogen into forms that other organisms can use, feeding plankton growth and supporting the food chain. Others perform denitrification, returning nitrogen gas to the atmosphere and maintaining equilibrium. Sulfate-reducing bacteria regulate sulfur levels, while methanotrophs consume methane before it escapes into the air. Every element of the ocean's composition passes through microbial hands in an endless cycle. The ocean's viral population, often overlooked, plays a surprising role in this balance. Trillions of viruses infect and destroy bacteria each day, releasing organic matter back into the water and fueling the microbial food web. Far from being purely destructive, these viral interactions control population sizes and genetic exchange, ensuring constant renewal. The sea's invisible warfare is also its greatest engine of stability. Oceanographers studying plankton blooms or climate change now understand that the key players are microscopic. Satellite images showing vast swirls of color, turquoise, green, or milky white, record the growth and death of microbial communities spanning hundreds of kilometers. Each bloom affects the carbon balance of the atmosphere, the acidity of seawater, and even global temperature. The Earth's climate, in many ways, is the result of microbial negotiation across the oceans. Marine snow, the steady rain of organic particles falling through the water column, connects all these layers. As it drifts downward, it feeds bacteria at every depth, forming a conveyor belt of energy that reaches the ocean floor. There, in the dark sediments, ancient microbes continue to metabolize carbon that may have begun its journey thousands of years before. From space, Earth appears as a blue sphere, yet the color we see is not only light reflected from water, it is also the signature of microbial activity. Billions of years after life began in the sea, microbes still dominate it, turning light and chemistry into movement and balance. Every wave, every current carries their quiet presence. Though they are unseen, their influence touches every breath of air above and every living thing below. Beneath every forest, field, and garden lies a world as rich and complex as any jungle above. The soil, which seems inert at first glance, is alive with microbial activity. Each handful contains billions of organisms, bacteria, archaea, fungi, and microscopic animals, all engaged in the slow, essential work of decomposition and renewal. It is one of the planet's most dynamic ecosystems, hidden in plain sight. Soil microbes are the silent architects of fertility. They break down the remains of plants and animals, converting complex organic matter into simpler molecules that can be absorbed again by roots and recycled through the food chain. Carbon compounds that once formed leaves, wood, or muscle become carbon dioxide, water, and nutrients such as nitrate and phosphate. This constant recycling maintains the thin layer of life-supporting material that blankets the continents. Without microbial decomposition, organic matter would simply accumulate, locking away the building blocks of new growth. One of the most vital processes in soil is nitrogen fixation. Although nitrogen makes up most of the atmosphere, it exists in a form that plants cannot use. Certain bacteria, known as diazotrophs, solve this problem by converting atmospheric nitrogen gas into ammonia through a complex enzyme system called nitrogenase. Some of these bacteria live freely in the soil, while others form partnerships with plants. Inside the root nodules of legumes such as peas and clover, they create miniature chemical factories where nitrogen is transformed and shared. The green of every leaf, the yield of every crop, depends on this invisible collaboration. Fungal networks also rely on bacteria to maintain the soil's delicate chemistry. Mycorrhizal fungi extend the reach of plant roots, trading nutrients with bacterial communities in exchange for carbon from the host plant. This underground web connects trees and grasses across vast distances, moving sugars, minerals, and even chemical signals. The entire forest becomes a single, interconnected organism sustained by microbial negotiation. Soil bacteria also control the flow of carbon through respiration. They consume decaying matter and release carbon dioxide back into the atmosphere, regulating global carbon levels. Some species form biofilms on soil particles, stabilizing structure and preventing erosion. Others produce sticky substances that bind soil grains together, improving its ability to retain water and air. These microscopic actions give rise to the texture and fertility that farmers depend on. In addition to nourishing plants, soil microbes act as guardians against disease. They compete with pathogens for space and resources, produce natural antibiotics, and trigger plants to strengthen their own immune responses. Many of the medicines humans use today, including streptomycin and tetracycline, were discovered in soil-dwelling bacteria. The ground beneath our feet is not only the foundation of agriculture but also a reservoir of biochemical invention. The diversity of the soil microbiome changes with every environment. In deserts, specialized microbes withstand dehydration by producing protective sugars that prevent cell collapse. In peat bogs, anaerobic bacteria slowly decompose organic matter, generating methane that escapes to the air. In tundra, cold-adapted species awaken briefly each summer to continue the slow recycling of frozen vegetation. Even in polluted or disturbed soils, microbial communities reorganize themselves, evolving to tolerate new conditions or metabolize synthetic chemicals. To study soil microbes is to witness the balance between decay and creation. Death feeds life, breakdown builds renewal. Every forest thrives because the soil beneath it never sleeps, always converting the old into the foundation for the new. Though we see only the visible plants and animals, it is the microbial multitudes below that keep the cycle of fertility turning, molecule by molecule, century after century. Life, once dominated by solitary single cells, soon discovered the power of cooperation. Microbes learned to live together, trading nutrients, shelter, and protection in partnerships that made survival easier for both sides. This principle, known as symbiosis, lies at the heart of all complex life. Every organism on Earth, from coral reef to human being, is built upon alliances first forged between microbes. The earliest symbiotic relationships appeared in shallow marine mats, where bacteria of different species grew side by side. One might capture sunlight, another might fix nitrogen, and another might remove toxic byproducts. Together they created stable communities that could withstand fluctuations of temperature, salinity, and light. These partnerships turned chaos into order, linking individual metabolisms into shared ecosystems. A more dramatic form of cooperation changed evolution itself, the union between ancient bacteria and primitive host cells that produced endosymbiosis. Around 2 billion years ago, a large archaeal cell engulfed a smaller bacterium, not as food but as a partner. The smaller cell provided energy through oxygen-based metabolism, while the host offered protection. Over time, the two became inseparable. The internal bacterium evolved into the mitochondrion, the energy-producing organelle found in nearly every complex cell today. A similar event later gave rise to chloroplasts, turning cells into photosynthetic engines. Every breath you take and every leaf that grows traces back to those ancient microbial mergers. Symbiosis, however, is not limited to ancient history. It continues everywhere. In the ocean, coral polyps house algae that feed them through photosynthesis, building entire reefs from calcium carbonate. In the roots of plants, bacteria and fungi exchange minerals for sugars, forming nutrient networks that keep forests alive. In animal intestines, microbes digest food and synthesize vitamins that the host cannot produce alone. Even insects depend on bacterial allies to supply essential amino acids missing from their diet. Each partnership follows the same basic principle. What one organism discards, another needs. Waste becomes resource. Byproduct becomes opportunity. Through this recycling, ecosystems achieve balance. In evolutionary terms, cooperation proves as powerful a driver as competition. The most successful species are not necessarily the strongest or the fastest, but those able to form lasting alliances. Scientists studying symbiosis have discovered that communication between partners occurs through chemical signals. Molecules released by one species trigger responses in another, coordinating activity across species boundaries. This biochemical conversation keeps relationships stable and responsive to change. In a coral reef, for example, heat stress can disrupt signaling and cause algae to leave their coral hosts, leading to bleaching. The health of the partnership depends on dialogue maintained at the molecular level. Symbiosis also shapes evolution by transferring genes. In some cases, partners share fragments of DNA, gradually merging their genetic identities. The mitochondria inside human cells still contain traces of their bacterial ancestry. This genetic blending blurs the line between separate organisms, revealing that life is less a collection of individuals and more a network of mutual dependencies. To look at nature through this lens is to see connection everywhere. The air we breathe, the food we eat, the energy we use, all exist because microbes learned to cooperate long ago. Even human civilization, with its agriculture, fermentation, and medicine, relies on microbial partners at every stage. The world's stability rests on the same principle that sustains a coral reef or a root nodule, life supporting life. In the stillness of the natural world, symbiosis is its quiet law. Every living system, visible or unseen, carries the imprint of microbial partnership. The smallest cells found strength not in isolation but in collaboration, proving that the essence of survival is shared purpose, a truth that continues to hold the planet together. Every person carries within them a living universe. Trillions of microorganisms inhabit the human body, forming a vast and intricate community known as the microbiome. These organisms, mostly bacteria, but also archaea, fungi, and viruses, outnumber human cells by at least 1-to-1. They are not invaders. They are long-term residents, partners in nearly every physiological process. From the surface of the skin to the depths of the intestines, they quietly sustain the balance that makes health possible. The human microbiome begins to form at birth. As an infant passes through the birth canal and takes its first breath, it encounters microbes that colonize the skin, mouth, and gut. Every touch, every sip of milk, and every interaction with the environment adds new species to the growing collection. Within days, the body becomes home to thousands of varieties, each adapted to a specific environment. Some prefer oxygen, others cannot tolerate it. Some feed on fats, others on sugars or fiber. Together, they create a self-regulating ecosystem that functions as an extension of the body itself. The gut microbiota is the most studied and perhaps the most vital. It helps digest complex carbohydrates, producing short-chain fatty acids that feed the cells lining the intestine. It synthesizes vitamins such as K and B12, breaks down toxins, and trains the immune system to recognize friend from foe. Research shows that people with a diverse gut microbiome tend to have stronger immune responses, better metabolism, and greater resistance to inflammation. The body's well-being depends not only on human genetics, but on this cooperative genetic archive of microbial life. Different regions of the body support distinct microbial communities. The skin hosts species adapted to dryness and exposure to light. The mouth contains biofilms that regulate acidity and prevent harmful bacteria from taking over. The lungs, once thought sterile, harbor low levels of bacteria that may help maintain immune balance. Even the eyes contain microbial traces that influence tear chemistry and defense mechanisms. Each habitat is unique, yet all are linked by the body's internal networks. Communication between host and microbes is constant. Cells of the immune system monitor bacterial signals, responding to their presence not with hostility but with negotiation. The microbes, in turn, release chemical messengers that influence digestion, hormone production, and even mood. Studies have revealed a gut-brain axis, where bacterial metabolites affect neurotransmitters like serotonin and dopamine. The phrase gut feeling may have deeper biological roots than once imagined. Modern life can disturb this delicate balance. Diets low in fiber, overuse of antibiotics, and chronic stress reduce microbial diversity, allowing harmful species to dominate. This state, called dysbiosis, has been linked to conditions ranging from allergies and obesity to anxiety and depression. Restoring equilibrium requires more than killing pathogens. It means nurturing the microbial partners that keep the system whole. Foods rich in natural fibers and fermented products, such as yogurt, kefir, and kimchi, help rebuild healthy communities. In recent years, scientists have begun exploring microbiome therapy, using probiotics or even microbial transplants to restore balance. Early results suggest that by adjusting our inner ecosystems, we can influence immunity, digestion, and emotional well-being. The body, it seems, is not a single organism but a consortium of thousands, bound by shared chemistry and mutual need. As you breathe now, consider that your own body carries the same bacterial legacy that once filled the early seas. Each cell of your microbiome traces its ancestry back billions of years, still performing the same simple acts of metabolism and exchange. You may notice your breathing settle into a steady rhythm, the kind that mirrors the quiet cooperation within. Even in rest, your inner ecosystems continue their work, silent, tireless, and perfectly synchronized with the life of the planet itself. The relationship between humans and their microbes is nowhere more apparent than in the digestive system. Stretching from mouth to colon, it is both a nutrient processing engine and a vast microbial habitat. Within it live hundreds of bacterial species that transform food into energy, regulate the immune system, and communicate with nearly every organ. This inner network functions with the precision of a coordinated ecosystem, maintaining the balance between nourishment and defense. When food enters the body, digestion begins through chemistry as much as biology. Saliva contains enzymes that start breaking down starches, but microbes quickly join the process. In the stomach, acid eliminates many foreign bacteria, yet some specialized microbes, such as Helicobacter pylori, have evolved to survive even there, adapting to the extreme acidity. Further along, in the small intestine, enzymes from the pancreas and liver continue dismantling fats and proteins into molecules small enough to absorb. Still, it is in the large intestine that microbes take the leading role. There, bacteria ferment complex carbohydrates that human enzymes cannot digest. This fermentation produces short-chain fatty acids, acetate, propionate, and butyrate, that provide energy to intestinal cells and influence metabolism throughout the body. These molecules also help maintain the barrier between the gut and bloodstream, preventing harmful substances from entering circulation. In this way, microbes not only process what you eat but also reinforce the body's physical and chemical defenses. The immune system and the microbiome are deeply intertwined. The gut contains more immune cells than any other part of the body. These cells constantly interact with microbial communities, learning which species are safe and which are dangerous. Early in life, these encounters train the immune system, teaching it tolerance and preventing unnecessary inflammation. Without exposure to harmless microbes, immunity becomes hypersensitive, overreacting to pollen, dust, or food. One reason why allergies and autoimmune diseases are more common in overly sterile environments. Certain bacteria also act as peacekeepers, releasing anti-inflammatory compounds that calm immune activity. Others compete directly with pathogens by occupying space on the intestinal lining or by producing antimicrobial peptides that keep invaders away. When antibiotics wipe out large portions of gut flora, harmful bacteria can seize the opportunity to grow unchecked, leading to infections such as Clostridioides difficile. Maintaining microbial diversity is therefore essential for long-term resilience. Nutrition, stress, and sleep all influence this balance. Diets rich in fiber, vegetables, and fermented foods encourage beneficial species, while processed foods and chronic stress disrupt their stability. Exercise and adequate rest strengthen the gut-immune connection by improving circulation and reducing inflammation. The system works best not under constant vigilance, but through quiet cooperation between host and microbe. Scientists have begun to explore how gut microbes influence diseases far beyond the digestive tract. Evidence links them to diabetes, heart health, and even neurological disorders. Some species affect cholesterol metabolism. Others produce molecules that alter immune signaling throughout the body. The idea that intestinal bacteria might influence mood or cognition once seemed implausible, but the gut-brain axis now forms one of biology's most active research fields. What emerges from all this study is a picture of unity. The immune system is not a separate guardian defending against all microbes. It is a mediator that fosters collaboration while controlling threats. Your health depends on this negotiation, an ancient, continuous dialogue between human cells and microbial partners. When viewed in this way, the act of digestion becomes more than a mechanical process. It is a daily renewal of an alliance that began in the earliest oceans, when bacteria first learned to exchange energy and information. Every meal continues that conversation, every heartbeat echoes it, and every breath carries the outcome of billions of quiet agreements that keep you alive. Health, at its most fundamental level, is balance, a dynamic state of cooperation between the human body and its microbial inhabitants. When this equilibrium holds, energy flows efficiently, immunity remains calm, and inflammation stays under control. But when it falters, even slightly, the results can ripple throughout every system of the body. Microbes thrive on stability, yet they are also the first to respond when that stability is disturbed. Inside the gut, thousands of bacterial species coexist in a fragile hierarchy. Beneficial microbes produce vitamins, digest fiber, and create acids that keep harmful organisms in check. Others remain neutral, waiting for opportunities to grow if space or nutrients become available. The key to balance lies not in eliminating microbes but in preserving diversity. When one species dominates, the ecosystem loses resilience. This condition, known as dysbiosis, can result from antibiotics, infection, poor diet, or chronic stress. Its effects reach far beyond digestion, into metabolism, immunity, and even mood regulation. A healthy microbiome acts as a shield, protecting its host through colonization resistance. Good bacteria occupy available surfaces in the intestine, consume nutrients that pathogens need, and secrete natural antibiotics that discourage invasion. In this competitive environment, harmful species struggle to gain a foothold. When the community is disrupted, pathogens such as Salmonella or Clostridium difficile can multiply unchecked, releasing toxins that damage tissues and provoke immune overreactions. The immune system, in turn, strives to restore balance. Specialized cells monitor microbial populations, responding with precision rather than aggression. They release signals that increase mucus production, recruit beneficial species, or suppress inflammation where needed. It is a delicate feedback loop. The microbiome teaches the immune system restraint, and the immune system ensures the microbiome's boundaries remain intact. When this conversation breaks down, the result can be chronic inflammation, allergies, or autoimmune disorders, signs that communication between the two sides has faltered. Diet is one of the most powerful influences on this equilibrium. Fibers from fruits, vegetables, and grains feed microbes that produce short-chain fatty acids, molecules known to strengthen the intestinal barrier and regulate immunity. Processed foods and excess sugar, by contrast, reduce microbial diversity and promote inflammation. Even small dietary changes can alter the composition of the gut within days, demonstrating how responsive this ecosystem is to its environment. Lifestyle plays a role as well. Physical activity stimulates beneficial bacterial growth by improving blood flow and intestinal motility. Stress, on the other hand, releases hormones that change gut chemistry, affecting which species thrive. Sleep quality, hydration, and exposure to nature all subtly shape the microbial community. Every choice, from diet to routine, participates in a continuous negotiation with the organisms that inhabit you. The search for balance extends beyond the gut. On the skin, microbial communities prevent infections and maintain the body's barrier to the outside world. In the lungs, they regulate immune tone, reducing the risk of inflammatory disease. Even in the reproductive tract, microbial diversity protects against pathogens and supports fertility. Health, viewed through this microbial lens, becomes a shared condition rather than an individual one. Medical science increasingly acknowledges this partnership. Treatments once focused solely on eradicating microbes now aim to restore ecological harmony. Probiotics and prebiotics, though still under study, reflect this shift from warfare to balance, an effort to support the allies within rather than destroy the ecosystem outright. The goal is not sterility but stability, not absence of microbes but presence of the right ones. When harmony holds, the microbiome works in quiet synchrony with its host. Energy flows smoothly, defense systems remain alert yet peaceful, and each part of the body communicates with the rest through unseen molecular channels. The lesson of health is the same lesson written across all of life's history. Survival depends not on domination but on cooperation, the steady balance between complexity and order that life itself has always pursued. For all the harmony life can achieve, nature also allows for conflict. Within the microbial world, not every relationship is cooperative. Some bacteria evolved to exploit the bodies of others, turning the same skills that once built balance into tools of invasion. These are the pathogens, organisms that cause disease by feeding on or damaging their hosts. Yet even their existence reflects the broader logic of evolution. Adaptation, competition, and survival in changing environments. The earliest pathogens likely emerged from ordinary microbes that lost their stable habitats. When conditions shifted or food became scarce, they turned to other living organisms as new sources of energy. Over generations, random mutations refined their ability to attach to host cells, evade immune defenses, and extract nutrients without immediately killing their hosts. Disease, in this sense, is an unintended byproduct of microbial ingenuity, a delicate negotiation that sometimes fails. Many pathogens rely on virulence factors, specialized molecules that help them breach barriers or disable defenses. Vibrio cholerae, the bacterium responsible for cholera, produces a toxin that disrupts water balance in intestinal cells, causing dehydration and rapid fluid loss. Mycobacterium tuberculosis hides within immune cells themselves, using the body's own defenses as camouflage. Streptococcus pneumoniae cloaks its surface with sugars to evade recognition, while Staphylococcus aureus produces enzymes that dissolve tissue, allowing it to spread. Each strategy reflects a deep understanding of host biology, evolved through countless generations of trial and error. For humans, the appearance of pathogens shaped history as profoundly as any geological or political event. Epidemics guided the movement of populations, the rise and fall of cities, and the development of medicine. Yet, on a microscopic level, infection is not a war in the traditional sense. It is a conversation between host and invader, each adapting to the other. The immune system responds with inflammation, fever, and antibodies. The pathogen counters by mutating, disguising, or retreating into dormancy. When balance is restored, the host recovers. When it fails, disease takes hold. Pathogenic evolution moves fast. Bacteria reproduce in minutes, and each generation carries the potential for genetic change. Through horizontal gene transfer, they can exchange plasmids, small DNA loops that carry instructions for toxins or antibiotic resistance. This sharing allows even unrelated species to acquire new abilities almost instantly. It is one reason infections can spread and adapt faster than medicine can respond. Still, the line between pathogen and ally is not fixed. Many microbes that are harmless or even beneficial can become dangerous under the right conditions. When the immune system weakens or the microbial community loses balance, once friendly bacteria may switch roles. Escherichia coli, a normal resident of the gut, can cause severe illness if it enters the bloodstream or acquires virulence genes. Such flexibility shows that disease is less about malice than about opportunity. Human progress has always depended on understanding and managing this delicate relationship. The development of sanitation, vaccines, and antibiotics dramatically reduced deaths from infection, but the underlying biological contest continues. Each advance creates new selective pressures that pathogens eventually learn to overcome. Antibiotic resistance, now a global concern, is simply evolution playing out at human speed. Yet even in their danger, pathogens reveal life's remarkable inventiveness. They show how quickly biology can adapt, how microscopic forms can outthink the largest organisms by sheer speed and variation. Studying them has led to vaccines, immune therapies, and genetic technologies that save millions of lives. The same mechanisms that cause harm have inspired cures, turning conflict into knowledge. In the end, pathogens remind us that the story of life is not one of perfect peace. It is an ongoing negotiation between creation and destruction, cooperation and competition. Disease, though feared, is part of the same evolutionary fabric that produced health itself, a shadow cast by life's ceaseless drive to persist, change, and survive. As human societies grew larger and more connected, microbes found new and fertile ground. Villages became towns, towns became cities, and for the first time, vast numbers of people lived close together. Along with food, trade, and culture, they shared something else: disease. Pathogens that had once burned out in small populations could now circulate indefinitely, moving from one host to another without pause. Thus began the age of epidemics, when the invisible world of microbes began shaping the course of human history. The first great epidemics left marks that archaeology still uncovers: mass graves, disrupted settlements, sudden declines in trade. The agents behind them were simple in form yet complex in behavior. Some came from animals, crossing species barriers through shared water, soil, or contact. Zoonotic transfer, the leap of a pathogen from animal to human, created waves of illness that changed civilizations. Smallpox, measles, and influenza all began as animal diseases before adapting to human hosts. One of the most infamous was the Black Death, caused by Yersinia pestis, which ravaged Europe in the 14th century. Spread by fleas that infested rats, it killed tens of millions and reshaped economies, labor systems, and belief structures. But long before that, ancient empires faced similar devastation. Smallpox in Egypt, typhus in Greece, and plague in Byzantium. Each outbreak forced societies to reorganize, advancing sanitation, medicine, and public health. Disease, in its cruelty, also became a teacher. Microbes exploit connection, and as exploration and trade expanded, so did their reach. Ships carried not only spices and silver, but also pathogens across oceans. In the wake of colonization, smallpox and measles decimated indigenous populations in the Americas, where immune systems had never encountered such infections. The microbial world, indifferent to borders or morality, reminded humanity that every movement and exchange carried risk as well as opportunity. Yet these same encounters led to progress. The study of contagion inspired observation, record-keeping, and experimentation. By the 17th century, physicians began to see patterns rather than curses. Microscopes revealed tiny organisms in water droplets. Experiments with cowpox led to the first vaccines. In the 19th century, scientists like Pasteur and Koch demonstrated that specific microbes caused specific diseases, transforming medicine into a science of prevention. The discovery of antibiotics in the 20th century gave humanity its most powerful weapon, though, as later generations would learn, not an eternal one. Epidemics are as much social phenomena as biological ones. Fear spreads alongside infection, changing how communities interact. Quarantines, masks, and isolation are ancient responses, recurring with each new outbreak. Modern transportation now allows a pathogen to circle the globe in days, yet our defenses, hygiene, vaccination, cooperation follow the same ancient logic: limit transmission, strengthen immunity, protect the vulnerable. The lessons of each epidemic have built upon the last. Sanitation transformed cities. Vaccination nearly eradicated smallpox. Surveillance and genomic sequencing now track new viruses in real time. Each advance draws from centuries of suffering and observation. The microbes evolve, but so do we. And yet, even with all this progress, the relationship remains familiar. Pathogens remind humanity that it is part of nature, not above it. Every outbreak exposes how deeply interconnected all living systems are, how air, water, and contact link every person to the microbial web of the planet. As you breathe quietly, the air around you carries descendants of those same microorganisms that once traveled across continents. The rhythm of respiration, oxygen entering and carbon dioxide leaving, is the same pattern that links you to them. You may notice your breathing become calm and even a reflection of the balance that life always seeks to restore after turmoil. Through every epidemic and every recovery, that rhythm endures, steady, patient, and timeless. For most of human history, infection was a constant shadow. A minor wound or cough could turn fatal, and even the best physicians could offer little more than comfort. That changed dramatically in the 20th century when science uncovered one of nature's own secrets: the ability of microbes to fight each other. From that discovery came antibiotics, molecules that transformed medicine, saved millions of lives, and altered humanity's relationship with the microbial world. The story begins in 1928, when Alexander Fleming returned to his London laboratory after a holiday and noticed something unusual. A mold, later identified as Penicillium notatum, had contaminated one of his petri dishes and killed the bacteria growing nearby. The mold released a substance that prevented bacterial growth. Fleming named it penicillin. Though the idea of one microbe producing compounds to inhibit another was not new, his observation launched a revolution. By the 1940s, industrial production of penicillin had begun, turning what was once a scientific curiosity into a weapon against infection. Soon, chemists and microbiologists discovered dozens of similar substances, many derived from soil bacteria. Streptomycin, tetracycline, erythromycin, and others followed, each targeting a different bacterial process: cell wall synthesis, protein production, or DNA replication. Diseases that once filled hospitals with the dying became treatable. Wounds healed without infection. Pneumonia and tuberculosis lost their fatal inevitability. Antibiotics became symbols of progress, proof that humanity could master the unseen world. Yet evolution never rests. Every antibiotic exerts selective pressure, favoring the rare bacteria with mutations that allow them to survive. These survivors multiply, spreading resistance genes through entire populations. Bacteria possess an extraordinary ability to share these genes horizontally, passing them even to unrelated species. Within decades, strains that were once easily defeated began to reappear, immune to multiple drugs. The earliest warnings came from hospitals, where resistant staphylococcal infections grew difficult to control. Later, bacteria carrying beta-lactamase enzymes began breaking down penicillin itself. In the following years, resistance expanded to nearly every class of antibiotic. The rise of multi-resistant pathogens such as MRSA, methicillin-resistant Staphylococcus aureus, and Klebsiella pneumoniae turned medicine's triumph into a renewed struggle. Each new antibiotic brought temporary relief before evolution inevitably caught up. Overuse and misuse have accelerated this process. Antibiotics prescribed for viral infections, incomplete courses of treatment, and widespread use in agriculture have all contributed to the spread of resistance. In many environments, traces of these drugs linger in water and soil, creating low-level exposure that drives mutation and adaptation. What began as a miracle of medicine now demands careful stewardship. Scientists are responding with the same creativity that marked the antibiotic era's beginning. They are searching for new molecules in extreme environments, deep-sea sediments, deserts, and caves, where microbes produce unique chemicals to compete for survival. Advances in genomics allow researchers to scan bacterial DNA directly for potential antibiotic genes. Others are revisiting an older concept, phage therapy, using viruses that infect and kill specific bacteria. Still, more researchers are exploring ways to disable resistance mechanisms rather than destroy the bacteria outright, turning pathogens back into manageable neighbors. The battle with microbial resistance is not a war with an enemy, but a dialogue with evolution. It reminds us that antibiotics are not weapons of conquest, but tools borrowed from nature, part of the microbial arms race that has existed for billions of years. To use them wisely is to acknowledge that we are participants, not rulers, in that ongoing process. The lesson of antibiotics and resistance is one of humility. Every advance carries its own consequences. Every victory invites adaptation. The smallest organisms continue to teach the largest that survival depends not on dominance, but on understanding. The power to heal, like the power to harm, resides in the same microscopic world that created both disease and its cure. For all the damage some microbes can cause, the vast majority are harmless or essential. Long before humans understood them, bacteria and fungi were already partners in daily life, quietly shaping food, drink, and medicine. Their chemistry preserved harvests, created nourishment, and produced flavors that became part of culture itself. To study beneficial microbes is to see how life's smallest forms have always worked alongside us, refining nature into art. The story of fermentation reaches back thousands of years. Ancient civilizations discovered that when fruit juice or grain mash was left exposed to air, it changed. Yeasts and bacteria transformed sugars into alcohol or acid, making products that lasted longer and tasted richer. The process was both preservation and creation. Bread, beer, cheese, yogurt, soy sauce, and wine, all are products of microbial craftsmanship. Before refrigeration or antibiotics, fermentation was humanity's most reliable form of food safety. Each fermented food relies on a specific community of microbes. In bread dough, yeasts such as Saccharomyces cerevisiae release carbon dioxide that makes the dough rise, while lactic acid bacteria add subtle tang. In cheesemaking, bacteria and molds break down milk proteins and fats, generating the complex aromas of cheddar, brie, or blue cheese. Yogurt depends on lactobacillus and Streptococcus thermophilus, which convert lactose into lactic acid, thickening the milk and creating its characteristic tartness. These transformations are not accidents, but the result of precise biochemical teamwork perfected over generations. Fermentation also plays a key role in nutrition. Microbes enhance the digestibility of foods and increase vitamin content. In sauerkraut and kimchi, fermentation boosts levels of vitamin C and beneficial probiotics that support gut health. In soy products like miso or tempeh, bacteria and fungi break down complex plant compounds, releasing amino acids and antioxidants. By pre-digesting nutrients, microbes extend the efficiency of human metabolism. The same microbial ingenuity underlies biotechnology. Long before genetic engineering, people used fermentation to produce vinegar, leavened bread, and alcoholic beverages. Today, those same principles power industrial processes that create enzymes, biofuels, and pharmaceuticals. Modern fermenters, operating under sterile conditions, harness bacteria and yeast to produce insulin, vitamins, and even biodegradable plastics. The line between ancient craft and modern science is thin. Both depend on the reliable metabolism of microorganisms. Beyond food and medicine, beneficial microbes sustain entire ecosystems. Nitrogen-fixing bacteria enrich soil, algae, and cyanobacteria, supply oxygen to aquatic environments, and decomposers return nutrients to circulation. In each case, life builds upon life, forming cycles of exchange that keep the planet stable. Humanity merely joins these cycles, borrowing what microbes already mastered. Their contribution extends to art, architecture, and climate. Certain bacteria produce pigments that color traditional textiles and paints. Microbes in limestone contribute to the slow formation of stalactites and caves. Others consume methane, a greenhouse gas, helping regulate Earth's temperature. Everywhere one looks, microbial labor underpins the balance of nature and the persistence of culture. When we understand this, gratitude replaces fear. The microscopic world is not hostile but generous. It feeds, protects, and teaches by example. Efficiency without waste, cooperation without noise, creation without recognition. As you listen quietly, think of these invisible artisans at work around you. In the air, in the soil, and within your body, they continue their patient transformations. Each breath carries molecules they once shaped, each heartbeat powered by their chemistry. You may notice your breathing becoming calm and natural, the mind settling into the same rhythm that governs their quiet persistence. Just as fermentation takes time and stillness to create something new, your own body renews itself in silence. Life's smallest workers continue their work, steady and tireless, allowing the world, and you, to rest. For most of history, humans used microbes without truly understanding them. Bread rose, wine fermented, wounds healed with moldy poultices, all by observation and luck. The 20th century transformed that relationship into deliberate design. Science learned not only to grow microbes, but to read and rewrite their genetic instructions. Out of that shift came biotechnology, the purposeful use of living systems to solve human problems. At its core, biotechnology harnesses microbial metabolism. Microbes are efficient chemical factories. They convert raw materials into complex products under mild conditions, often with little waste. Long before the term existed, brewing, cheesemaking, and vinegar production were examples of controlled microbial engineering. Modern biotechnology extends the same principle to medicine, agriculture, and environmental management, guided by genetic insight rather than intuition. The breakthrough moment arrived with the discovery of DNA as the carrier of genetic information and the realization that it could be cut, copied, and transferred between organisms. In the 1970 seconds, scientists used bacteria to produce human insulin, marking the first time a gene from one species was successfully expressed in another. The result was not only a life-saving therapy for millions with diabetes, but also proof that microbes could serve as programmable platforms for making almost anything life could design. Since then, microbes have produced a vast array of materials once derived from animals or extracted from nature in unsustainable ways. Engineered yeasts now manufacture vitamins, growth hormones, and vaccines. Bacteria create biodegradable plastics, enzymes for detergents, and amino acids for food supplements. The pharmaceutical industry relies on microbial cell cultures to produce antibiotics, anti-cancer drugs, and antibodies. In agriculture, biofertilizers replace chemical ones, and biopesticides made from bacterial toxins protect crops without polluting soil or water. Perhaps the most revolutionary advance emerged from bacteria themselves, the discovery of CRISPR-Cas systems. These molecular tools, derived from bacterial immune defenses against viruses, allow scientists to edit DNA with unprecedented precision. What microbes once used to remember and destroy viral invaders has become humanity's most powerful instrument for genetic engineering. With CRISPR, scientists can correct mutations, design resistant crops, and explore therapies for inherited diseases, all inspired by the simple survival strategies of ancient bacteria. Microbial biotechnology also offers hope for environmental recovery. Certain species digest petroleum, clean toxic metals, or capture carbon dioxide from the atmosphere. Bioreactors filled with algae and bacteria can convert industrial waste gases into fuel or useful chemicals. In wastewater treatment, microbial communities remove contaminants while generating energy from organic matter. Life's smallest forms, guided by human intention, are now repairing the damage of industrial growth. Yet this progress carries ethical and ecological questions. Modifying life invites responsibility. The same flexibility that makes microbes useful also allows them to spread beyond control if mishandled. Laboratories follow strict biosafety rules, but the broader challenge is philosophical. Biotechnology asks humanity to become a co-designer of evolution. Used wisely, it can heal and sustain. Used carelessly, it can disrupt delicate balances built over billions of years. Still, the promise is undeniable. Microbes have always been nature's engineers, experimenting endlessly in every environment. Biotechnology simply listens more closely to what they already know. It translates microbial wisdom into practical form, into medicine, sustainable materials, and energy systems that mimic natural efficiency. The story of biotechnology is, ultimately, a story of partnership. The same cells that once built oxygen into the atmosphere and soil into fertility now help shape the future of human civilization. Their potential remains vast, limited only by our understanding and care. In their genetic code lies not just the memory of life's past, but the blueprint for its continuation, a reminder that progress and preservation need not be opposites when guided by respect for the smallest architects of the world. The same invisible organisms that build soil and sustain oceans also perform one of the planet's least glamorous yet most vital tasks: cleaning. From decomposing fallen leaves in a forest to purifying the water in a city, microbes serve as Earth's natural recyclers. They transform waste into nutrients, toxins into harmless compounds, and polluted water into something drinkable again. Without them, decay would stop, rivers would choke with organic debris, and ecosystems would collapse beneath their own excess. In nature, waste never truly exists. What one organism discards becomes the raw material for another. Microbes are the agents of this transformation. When plants and animals die, bacteria and fungi break down their remains, releasing carbon dioxide, water, and mineral nutrients that re-enter the cycle of life. This decomposition powers forests, grasslands, and oceans alike, maintaining the flow of energy that makes ecosystems self-sustaining. Human civilization, however, concentrates waste in ways that nature did not anticipate. Cities generate billions of liters of wastewater every day, filled with organic material, chemicals, and pathogens. For more than a century, engineers have relied on microbes to manage this load through biological treatment systems. The process begins in aeration tanks, where bacteria suspended in water feed on organic matter. They form small clusters known as activated sludge, consuming waste as food and multiplying as they clean. Air bubbles supply oxygen, keeping aerobic bacteria active while they oxidize carbon compounds into carbon dioxide and water. In the next stage, microbes settle out as sludge and are partly recycled to start the process again. Additional bacterial communities remove nitrogen and phosphorus, preventing algal blooms when the treated water returns to rivers or seas. The result is clear water, safe for discharge, and often cleaner than the bodies of water that receive it. This biological purification is faster, cheaper, and far more sustainable than any chemical alternative. Some bacteria thrive even in oxygen-free stages of treatment. Anaerobic digestion, used in many wastewater and agricultural systems, relies on microbes that decompose organic material in sealed tanks. As they break down the waste, they release biogas, a mixture of methane and carbon dioxide that can be captured and used as renewable energy. The remaining solid residue, rich in nutrients, becomes fertilizer. The entire process closes the loop, turning pollution into power and waste into resource. Microbes also play a critical role in cleaning environments beyond treatment plants. In bioremediation, certain bacterial species degrade oil spills, pesticides, and industrial contaminants. For example, Pseudomonas species can metabolize hydrocarbons from petroleum, while Geobacter can transform toxic metals into stable forms. These microbial strategies are being applied to restore polluted soil, groundwater, and even radioactive sites. In some cases, scientists have genetically enhanced bacteria to target specific pollutants, accelerating natural recovery without introducing harmful side effects. This microbial work extends to the smallest scales, in household septic systems, in compost heaps, and even inside landfills. Bacterial communities continue to recycle organic waste. Each environment supports a chain of specialists. One group breaks large molecules into smaller fragments, another consumes those fragments, and yet another converts the residues into gas or mineral compounds. It is the same sequence that has operated since life began, now employed to sustain modern societies. What stands out in all these systems is efficiency. Microbes accomplish complex chemical conversions at ambient temperature and pressure, powered only by the energy within the waste itself. No machines, no flames, no pollution, just living chemistry, endlessly patient and perfectly calibrated. By observing and partnering with these organisms, humans have learned that cleanliness does not require eradication but collaboration. Purity in nature is not the absence of life but the presence of balance. Beneath every clear stream and beneath every field lies a hidden workforce that never sleeps, turning what we discard into the foundation of tomorrow's growth. The global climate may seem governed by oceans, forests, and winds, but the true regulators of the planet's atmosphere are far smaller. Microbes, through their collective chemistry, manage the balance of gases that control temperature and weather. Every molecule of carbon dioxide, methane, or nitrous oxide in the air has, at some point, passed through microbial hands. These microscopic reactions determine how much heat the planet retains, how fertile the soil becomes, and how long ecosystems can endure. At the center of this process is carbon cycling, the continuous exchange of carbon between land, sea, and air. Microbes are its engines. Photosynthetic bacteria in oceans and soils capture carbon dioxide, using it to build sugars and organic matter. When they die, other microbes decompose their remains, returning carbon to the atmosphere as carbon dioxide or methane. This endless cycle controls the planet's temperature far more delicately than any single climate event. In the ocean, cyanobacteria and microalgae fix vast amounts of carbon through photosynthesis, forming the base of the marine food web. As they sink and decay, other bacteria process the remains in a cascade of recycling. Some of that carbon settles on the sea floor, locked away in sediment for thousands of years. The deep ocean thus acts as Earth's greatest carbon reservoir, managed by microbial activity alone. Without these invisible caretakers, carbon dioxide would accumulate unchecked, transforming the climate beyond recognition. On land, soil microbes perform a similar task. They decompose dead leaves and wood, releasing carbon dioxide, while simultaneously storing carbon within stable organic compounds. In healthy forests and grasslands, this balance remains steady. The carbon released by decay equals the carbon absorbed by growth. But when soils are disturbed, through deforestation, ploughing, or warming, microbial respiration increases, releasing stored carbon into the air. The same processes that sustain life can, under pressure, amplify climate change. Microbes also control the levels of methane, a greenhouse gas many times more potent than carbon dioxide. In wetlands, rice paddies, and animal digestive systems, methanogenic archaea produce methane as a byproduct of metabolism. In contrast, methanotrophic bacteria consume it, converting methane back into carbon dioxide before it escapes into the atmosphere. The balance between these two groups determines whether an ecosystem acts as a source or sink of greenhouse gases. A small shift in temperature, moisture, or nutrient availability can tip this equilibrium and alter global climate patterns. The same is true for nitrous oxide, another powerful greenhouse gas. Certain soil bacteria generate it during nitrogen cycling, especially when oxygen levels fluctuate. Agricultural runoff rich in fertilizer fuels these reactions, increasing emissions. Yet other bacteria perform denitrification, completing the cycle and returning nitrogen harmlessly to the air. Once again, climate stability rests on microbial mediation, responding to human influence with biochemical precision. Beyond their control of gases, microbes shape the planet's reflectivity and cloud formation. Marine bacteria release compounds like dimethyl sulfide, which rise into the atmosphere and form condensation nuclei for clouds. These clouds, in turn, influence temperature by reflecting sunlight. A shift in microbial population can subtly alter cloud cover, linking the smallest organisms to the grandest patterns of weather. As climate change accelerates, scientists are studying how these microscopic systems respond. Warming oceans alter plankton populations. Thawing permafrost awakens dormant microbes that release stored methane. Acidifying waters change the balance of bacterial species. Each reaction feeds into the next, forming feedback loops that can either stabilize or intensify warming. Understanding these processes is essential because microbes are not merely responders to climate, they are its architects. They regulate composition, temperature, and renewal with quiet persistence. To preserve the planet's stability, we must consider not only forests and fuels, but also the microbial engines beneath them. The air you breathe, the clouds above you, and the warmth that surrounds the Earth all depend on the metabolism of unseen life. In their silent chemistry, the climate finds its pulse, steady and unending, as it has since the first cell released its first breath of gas into an ancient sky. For billions of years, microbes have lived in close quarters, on rocks, in soil, in the thin film of moisture that coats every surface. To survive in such density, they developed an extraordinary ability: communication. Though they lack nerves or voices, bacteria coordinate behavior through chemical messages, exchanging information with precision and purpose. What appears chaotic under a microscope is, in fact, a vast network of signaling, negotiation, and cooperation. This form of interaction is called quorum sensing, a process by which bacteria gauge their population density and act collectively once a critical threshold is reached. Each cell releases signaling molecules, known as autoinducers, into its surroundings. As the community grows, the concentration of these molecules rises. When it reaches a specific level, the signal binds to receptors on the bacterial surface, triggering a synchronized response across the colony. In an instant, thousands of cells begin behaving as one. Through quorum sensing, microbes regulate crucial activities, forming biofilms, producing toxins, emitting light, or releasing enzymes. For example, the marine bacterium Vibrio fischeri lives symbiotically in the light organs of certain squids. When only a few bacterial cells are present, they remain dark, but once their numbers reach the quorum, they begin to glow collectively, creating bioluminescence that helps the squid camouflage against the moonlit sea. This partnership demonstrates the elegance of microbial communication, a dialogue between species that benefits both. In other environments, communication governs defense and competition. Biofilms, the structured communities that coat rocks, pipes, and teeth, form only when cells signal their readiness to cooperate. Within these films, bacteria share nutrients and exchange genetic material while shielding themselves from external threats. The film's architecture is complex, resembling a miniature city with channels for nutrient flow and waste removal. Every layer responds to chemical cues, maintaining order through invisible conversation. Microbes also use signals to coordinate aggression. Some species release molecules that trigger neighboring cells to produce antibiotics, ensuring that competitors are suppressed only when population density warrants it. Others detect signals from rival colonies and alter their metabolism to avoid direct conflict. In this way, quorum sensing prevents wasted energy and preserves balance, a lesson in restraint encoded at the molecular level. Communication extends beyond single species. In soil and the human gut alike, diverse microbial communities trade molecular messages across boundaries. Signals from one species can influence the behavior of many others, creating cooperative networks that regulate the entire ecosystem. These interactions shape nutrient cycles, influence plant growth, and even affect the immune responses of animals. Scientists are now learning to intercept and interpret these microbial conversations. By identifying the molecules used in quorum sensing, researchers can design drugs that block or mimic them. Such quorum-quenching strategies could one day prevent infections not by killing bacteria outright, but by silencing the signals that trigger harmful behavior. In agriculture, manipulating microbial communication might encourage beneficial bacteria to form protective biofilms around plant roots, improving growth without chemical fertilizers. The discovery of microbial signaling has transformed our view of evolution. Rather than isolated entities competing blindly, bacteria appear as social organisms capable of cooperation, altruism, and coordination. They respond to their environment as part of a collective intelligence, proving that communication is not the privilege of complex creatures but a universal feature of life. Every living cell, from the smallest bacterium to the largest whale, depends on chemical language. In the microbial world, this language is silent yet constant, composed of molecules diffusing through water and air. Its grammar is one of timing and concentration. Its meaning is survival. If we could listen to this unseen dialogue, we would hear the soft murmur of life negotiating its own harmony, an endless exchange of signals that has guided the planet since its earliest days. The world we see above ground, with its forests, oceans, and animals, is built upon this quiet conversation that never stops. In the microscopic world, harmony and conflict are two sides of the same coin. Microbes cooperate to build communities, share resources, and stabilize ecosystems. Yet they also compete fiercely for space and energy. Their survival depends on a balance between collaboration and rivalry, a dynamic that mirrors the larger patterns of nature and even human society. One of the most striking examples of microbial cooperation is the biofilm, a complex multicellular structure formed when individual cells attach to a surface and embed themselves in a matrix of secreted polymers. Biofilms grow on river stones, medical implants, and even teeth, forming plaque. Within these slim layers, bacteria coordinate through chemical signals, dividing labor and sharing nutrients. The outer layers protect against antibiotics and physical stress, while the inner cells focus on growth and repair. Together, they create a living fortress that can endure conditions lethal to solitary cells. Biofilms demonstrate a principle that evolution rewards. Unity increases resilience. Each bacterium contributes to the collective good, even though it could theoretically live alone. By pooling resources, they create microenvironments of stability, pockets of oxygen, gradients of nutrients, and channels for communication. Yet, even within cooperation, tension exists. Some bacteria exploit the benefits without contributing, taking advantage of the community's shared defenses. To maintain order, biofilms have developed internal regulation systems, favoring cells that cooperate and punishing those that do not. This balance between self-interest and group benefit is ancient, perhaps the earliest form of social contract on Earth. Competition among microbes is equally creative. Because resources are limited, bacteria have evolved intricate strategies to outmaneuver one another. Some release bacteriocins, protein weapons that kill closely related strains while sparing themselves. Others secrete toxins that dissolve cell membranes or disrupt DNA replication. Even oxygen, a molecule most organisms depend on, can become a weapon. Certain bacteria produce reactive oxygen species to damage competitors, using chemistry itself as a form of warfare. There are also quieter, subtler forms of rivalry. Microbes can occupy niches so completely that newcomers cannot establish themselves, a phenomenon called competitive exclusion. They may alter their surroundings, changing pH, or depleting nutrients to disadvantage rivals. These invisible battles take place everywhere, in soil, in oceans, and within the human body. The microbial world operates as a vast marketplace of adaptation, where every gain demands energy and every advantage is temporary. Yet from competition arises balance. No single species can dominate forever because cooperation always re-emerges to stabilize the system. In the human gut, for instance, hundreds of bacterial species coexist by dividing labor, one breaking down fiber, another consuming the byproducts, and a third converting waste into vitamins. This layered cooperation ensures that resources are fully used, leaving little room for pathogens to invade. The result is a dynamic equilibrium maintained not by peace, but by constant negotiation. Scientists studying these interactions often describe microbial communities as ecosystems in miniature. Their structure reflects the same principles that govern forests and coral reefs. Competition drives diversity, and cooperation sustains it. Microbes demonstrate that conflict and collaboration are not opposites but partners in evolution. Each enhances the other, shaping the adaptability of life as a whole. The molecules used in microbial warfare have given humans invaluable tools. Many antibiotics, including penicillin and streptomycin, originated as bacterial or fungal weapons refined through natural selection. What microbes designed to eliminate competitors, medicine now uses to save lives. Once again, human progress echoes microbial strategy, turning conflict into creation. In the end, the microbial world teaches that survival is rarely solitary. Even the fiercest competition occurs within a web of interdependence. Cooperation builds resilience, rivalry drives innovation. Together they form the rhythm of evolution, an ancient balance that underlies every living system, from a single cell to an entire biosphere. In the visible world, change seems slow. Mountains rise over millennia, forests shift over centuries, and species evolve over eons. Yet for microbes, evolution happens almost instantly. Their lives unfold in minutes, not decades, and each new generation carries the potential for mutation and adaptation. Through this rapid cycle, microbes reveal evolution not as an abstract theory, but as a living, observable process, a continuous rewriting of life's code in real time. Bacteria reproduce by simple division. One cell becomes two, two become four, and so on. Each division brings the chance of a genetic alteration, a small copying error in the DNA sequence. Most mutations do nothing or even harm the cell, but a few offer advantages: resistance to a chemical, tolerance to heat, or faster growth under limited nutrients. Those with beneficial mutations survive and multiply, passing their new traits to countless descendants. In hours, a population can adapt to a change that would take larger organisms thousands of years. This rapid adaptability explains why microbes thrive in every environment. When antibiotics are introduced, a few resistant cells inevitably survive, rebuild their numbers, and become dominant. When temperatures rise, microbes evolve enzymes that remain stable in heat. When nutrients run low, they learn to consume new sources of energy. Evolution is not a distant event in their history. It is the constant background rhythm of their existence. Another powerful engine of microbial evolution is horizontal gene transfer, the exchange of genetic material between unrelated cells. Unlike animals, which inherit genes only from their parents, bacteria can trade DNA fragments across species. They use tiny structures called pili to share plasmids, or they absorb stray genetic material from dead cells. In this way, a single useful gene, for antibiotic resistance or toxin production, can spread through a population in hours. The microbial world functions as a global genetic marketplace, where innovation circulates freely. Viruses accelerate this process further. Many bacterial viruses, called bacteriophages, inject their genetic material into cells and sometimes carry pieces of bacterial DNA with them when they move on. These exchanges create endless combinations, driving the diversification of life at a molecular level. Evolution, rather than being a slow climb up a branching tree, resembles a web of continuous exchange, a living network that constantly reconfigures itself. Researchers witness microbial evolution daily in the laboratory. Long-term experiments, such as those conducted by Richard Lenski with Escherichia coli, have tracked tens of thousands of generations. Over decades, these bacteria evolved new metabolic capabilities, including the ability to consume citrate, a compound they could not digest before. Each evolutionary step, recorded in DNA sequences, shows adaptation unfolding as a measurable process. Microbial evolution also plays a crucial role in ecology. In oceans and soils, microbes adjust their metabolism to shifts in temperature, light, or nutrient availability, ensuring that global cycles of carbon and nitrogen continue smoothly. Their adaptability keeps ecosystems resilient against change, turning what might seem chaotic into equilibrium. This constant transformation carries both promise and warning. It makes microbes indispensable to planetary balance, but also allows pathogens to evade treatment and climate systems to react unpredictably. Evolution, for microbes, is neither good nor bad. It simply is. It is the mechanism by which life persists in an ever-changing world. You might notice, as you breathe gently, that this rhythm of adaptation mirrors the rhythm of rest, slow, steady, and continuous. Just as each breath renews the body, each microbial generation renews life's potential. Even in stillness, change is happening all around you and within you, invisible but unceasing. The smallest organisms never stop evolving, quietly shaping the present and preparing the future, one division at a time. When life first appeared on Earth, it evolved under conditions that seemed harsh by human standards. Extreme heat, radiation, and bombardment from space. It is no surprise, then, that scientists have long wondered whether microbes could also survive beyond our planet. The question is not just whether life could exist elsewhere, but whether the most resilient organisms on Earth could endure the vast, hostile emptiness of space itself. Research has revealed that many microbes possess extraordinary endurance. Certain bacterial spores can remain viable after years of desiccation, freezing, and radiation exposure. These spores, surrounded by protective layers of protein and minerals, can effectively shut down metabolism, entering a suspended state that preserves DNA and enzymes. In this form, they resist ultraviolet light, vacuum, and temperature extremes, the very challenges that define space. Experiments conducted on spacecraft and space stations have tested this resilience directly. Microbes such as Bacillus subtilis and Deinococcus radiodurans have been exposed to open space for months and even years. When retrieved and rehydrated, many of them revived, reproducing as if nothing had happened. The findings suggest that life's chemistry is remarkably stable, capable of enduring radiation doses and vacuum conditions that would destroy most complex organisms. This durability has given rise to the idea of panspermia, the hypothesis that life might travel between planets on meteoroids or comets. Meteorite impacts can eject rocks from a planet's surface into space, and some of those rocks could carry microbial passengers. Protected within tiny cavities or layers of ice, microbes might remain dormant during interplanetary journeys, lasting millions of years, only to awaken when conditions become favorable again. Though unproven, panspermia offers a striking possibility that life did not start in one place, but spread wherever it could take hold. Space agencies have also found that microbes thrive in artificial habitats beyond Earth. On the International Space Station, bacterial colonies form biofilms on surfaces, demonstrating that even in microgravity, life organizes itself into communities. Some microbes become more virulent or resistant in space, adapting rapidly to low gravity and radiation stress. For astronauts, these findings highlight both the risks and benefits of microbial companionship. While contamination is a concern, microbes also help recycle air and waste, and may one day assist in producing food or medicine during long missions. Beyond survival, microbes offer practical tools for space exploration. Scientists envision using biotechnology in space to convert carbon dioxide into oxygen, create nutrients from waste, or extract minerals from extraterrestrial rock. Engineered microbes could help terraform future colonies or maintain closed-loop life support systems. Just as microbes once made Earth's atmosphere breathable, they might one day make other worlds habitable. These experiments also inform the search for life beyond our planet. By understanding how Earth's microbes respond to cosmic conditions, scientists refine their expectations for what might survive on Mars, Europa, or Enceladus. Evidence of methane, organic molecules, and possible subsurface water on those worlds strengthens the idea that microbial life could exist there, perhaps deep beneath ice or rock, sustained by chemical energy as it is near Earth's hydrothermal vents. Each discovery expands the definition of habitability. Life may not need sunlight, soil, or oxygen, only energy, stability, and time. Microbes demonstrate that biology is not fragile but adaptable, capable of rewriting itself to fit almost any environment. Whether they ever journeyed through space naturally or not, their resilience shows that the boundary between biology and the cosmos is thinner than once believed. From the deepest ocean trench to the vacuum beyond the atmosphere, the same pattern repeats. Wherever conditions allow molecules to interact, life finds a way. Microbes carry the legacy of that persistence. They may be Earth's smallest explorers, carrying the signature of our planet far beyond its borders, proving that life's reach is as vast and enduring as the universe itself. For centuries, scientists believed life existed only within a narrow band of conditions: moderate temperature, gentle pressure, and abundant sunlight. But as exploration deepened into the planet's extremes, that idea dissolved. Microbes have been found in boiling springs, frozen glaciers, acidic pools, and the deep crust of the Earth, thousands of meters below the surface. The more we search, the more it becomes clear. Life's boundaries are far wider than once imagined. In the scalding waters of Yellowstone and Kamchatka, extremophiles flourish at temperatures above 90 degrees Celsius. Their enzymes remain stable where most proteins unravel, thanks to tightly folded structures and specialized amino acids. These adaptations allow them to harvest energy directly from chemical reactions involving sulfur and iron. In the opposite extreme, psychrophiles thrive in Antarctic ice and Arctic permafrost. They produce antifreeze proteins that prevent ice crystals from piercing their membranes, remaining active even below freezing. Deep beneath the ocean floor, barophiles live under pressures 1,000 times greater than at sea level. Instead of breaking apart, their cell membranes become more flexible, their enzymes tuned to function in the crushing dark. Others, called halophiles, exist in salt concentrations so high that ordinary cells would shrivel from dehydration. They balance osmotic stress by filling themselves with potassium and protective solutes, maintaining equilibrium with the brine around them. Radiation-resistant microbes push endurance even further. Deinococcus radiodurans, often called the world's toughest bacterium, can survive doses of radiation that would fragment human DNA beyond repair. It accomplishes this by storing multiple copies of its genome and reassembling them like a molecular puzzle after damage occurs. Similar mechanisms protect it from desiccation, hinting that resistance to radiation evolved first as a defense against drought. In recent decades, scientists drilling into deep rock have discovered entire biospheres far from sunlight. Endolithic bacteria live within mineral pores, feeding on trace gases like hydrogen and methane. In these lightless realms, life depends entirely on geochemical energy. Some of these microbes replicate so slowly that a single generation may take centuries, yet they persist, proof that vitality does not depend on speed or abundance, only on continuity. Such discoveries have redefined the concept of habitability. The conditions that sustain microbes on Earth mirror those that may exist elsewhere, beneath Mars's surface, within Europa's subsurface ocean, or under the ice of Enceladus. If life can endure the crushing pressure of the deep crust or the desolation of a salt desert, it may also endure on other worlds. Astrobiologists studying these extremophiles see them as ambassadors of possibility, showing that life's chemistry is more universal than we once dared to think. Even within the human body, microbial resilience echoes this same principle. Certain bacteria in the gut survive shifts in diet, temperature, and pH with effortless precision. Their capacity to adapt to changing environments mirrors the endurance of microbes beneath glaciers or volcanic vents. The secret lies in flexibility, the ability to alter metabolism, structure, or gene expression in response to new stress. Life, from its smallest form, was designed not for comfort but for persistence. You may notice your breathing slow as you think of these extremes, of organisms thriving where no warmth, light, or comfort exists. Their existence reminds us that endurance does not always roar. Sometimes it whispers, waiting patiently through ages of stillness. Just as they survive frozen or compressed, you too can rest in stillness without losing life's spark. The same quiet resilience that keeps microbes alive in stone and ice continues within you, steady and unbroken through every cycle of change. From boiling springs to buried rock, from the vacuum of space to the rhythm of a heartbeat, the lesson is the same: life endures. Its limits are far beyond what we can see, and its persistence, silent and absolute, continues through every night, through every moment of rest. Though invisible to the eye, microbes form a vast and interconnected network that binds all living things together. Every plant, animal, and ecosystem depends on this hidden web of relationships. It links the soil to the atmosphere, the oceans to the clouds, and the living to the dead. Without microbes, the chain of life would break within days. They are the silent infrastructure of the biosphere, unseen engineers maintaining the balance that makes existence possible. In the soil beneath a forest, bacterial and fungal communities connect trees and grasses through the exchange of nutrients. Mycorrhizal fungi form filaments that extend from root to root, while bacteria on their surfaces help fix nitrogen or dissolve minerals. Together, they create networks that allow entire ecosystems to share resources. A tree shaded from sunlight may receive carbon compounds from its neighbors through these subterranean channels, sustained by microbial cooperation. The forest becomes a single living organism, its health dependent on the communication that microbes enable. In the oceans, microscopic plankton, cyanobacteria, and archaea perform the same role on a planetary scale. They link the atmosphere with the deep sea, recycling carbon and oxygen across thousands of kilometers. Each wave carries countless living cells that drift, divide, and die, releasing nutrients for others to use. The ocean's color, chemistry, and ability to store heat all depend on this continuous microbial exchange. Even clouds are seeded by bacteria swept upward from the sea's surface, influencing rainfall and weather. Animals, too, exist within this unseen web. Every insect, bird, and mammal carries its own microbial signature, a living community shaped by diet, habitat, and ancestry. When species interact, their microbes mix, forming temporary bridges between ecosystems. A bee transferring pollen also transfers bacteria that help plants resist disease. Migratory birds carry microbes across continents, spreading diversity and stability as they travel. The health of entire populations depends not only on genetic inheritance but on the microbes that accompany them through generations. Humans often imagine themselves apart from nature, yet our connection to the microbial web is constant. Breathing draws in bacteria and spores from the air. Eating invites new species into the gut. Touch transfers microbial traces between bodies and surfaces. Even the buildings we inhabit have microbial identities, shaped by those who live inside. Far from being sterile, our constructed spaces host miniature ecosystems that adapt to light, humidity, and human presence. When viewed from this perspective, life on Earth appears not as a collection of separate beings, but as a single organism composed of countless smaller ones. Each microbe plays a role in regulating energy, matter, and information. Disruptions in one part of the system ripple through the whole. Deforestation changes soil microbes, ocean warming alters plankton, pollution affects the microbes that clean air and water. The planetary web responds continuously, seeking equilibrium but never resting. Modern science, with its genomic tools and satellite sensors, is beginning to map this invisible network. Researchers can now trace microbial genes through the atmosphere, track their movement in ocean currents, and observe how they migrate across continents. This emerging field, microbial ecology on a planetary scale, shows that the biosphere operates as an integrated whole. Microbes are not merely background players, they are the circuitry of life itself. Recognizing this web carries a quiet lesson in perspective. Every breath, every drop of rain, every handful of soil contains a reflection of the same process, the exchange of life with life. The world's stability is not sustained by dominance or control, but by connection, the invisible cooperation of countless unseen partners. To understand microbes is to understand ourselves as part of something immeasurably larger. The web they weave holds not just ecosystems, but the memory of creation and the promise of renewal. In their silent work, they remind us that everything living, great or small, is linked by the same thread of existence, endlessly woven through time and space. The story of microbes is not only about the past or the present, it is also the blueprint for what comes next. As humanity faces climate change, pollution, food insecurity, and declining biodiversity, the smallest organisms may hold the most powerful solutions. Their chemistry, honed over billions of years, already performs the tasks humans now struggle to replicate: recycling waste, capturing carbon, producing energy, and restoring balance. The future of the planet may depend on how well we learn to collaborate with them rather than control them. In laboratories around the world, scientists are exploring synthetic biology, a field that designs and reprograms microbes to perform new functions. By modifying genetic sequences, researchers create bacteria that can detect toxins, clean contaminated water, or produce sustainable materials. Some engineered microbes already turn agricultural waste into biofuel. Others generate biodegradable plastics that break down without harming the environment. These innovations represent a new era in which biology becomes a form of engineering, one guided by natural efficiency rather than industrial excess. Microbes are also shaping the future of medicine. Advances in microbiome research suggest that personalized treatments could one day be built around each person's microbial profile. Instead of prescribing drugs that act broadly on the body, physicians might use targeted probiotics or microbial transplants to correct imbalances, enhance immunity, and even influence mental health. Already, fecal microbiota therapies are being used to treat chronic intestinal diseases, with results that often surpass traditional antibiotics. The frontier of healthcare is no longer chemical alone, but ecological, focused on restoring harmony rather than destroying invaders. In agriculture, microbes are becoming key allies in sustainable farming. Nitrogen-fixing bacteria and phosphate-solubilizing microbes reduce the need for chemical fertilizers, while others protect crops from pests by strengthening natural defenses. Researchers are developing microbial seed coatings that help plants grow in drought or poor soil, allowing food production to adapt to a changing climate. Instead of forcing growth through artificial inputs, future farming may rely on invisible networks of microbial support, the same partnerships that sustained ecosystems long before agriculture existed. Energy production, too, is entering a microbial phase. Certain bacteria produce electricity directly by transferring electrons to metal surfaces, creating microbial fuel cells that can power sensors and remote systems. Others generate hydrogen or methane from waste, offering renewable alternatives to fossil fuels. These living power plants could one day run on sunlight and organic matter, turning biology into the world's cleanest source of energy. Even construction and manufacturing may soon depend on microbial processes. Researchers are growing bioconcrete, in which bacteria repair cracks by depositing minerals. Other species produce pigments, fibers, or structural proteins for textiles and biodegradable packaging. By observing how microbes build shells, reefs, and films in nature, engineers are learning to design materials that self-heal, adapt, and return safely to the environment when no longer needed. This microbial future will not be without challenges. Ethical questions about genetic modification, ecological impact, and containment remain critical. Yet these concerns mirror those faced at every scientific threshold. The difference today is that we understand more clearly what is at stake, not simply human progress, but planetary stability. The closer we look at microbes, the more we see that they have always managed the tasks we are only beginning to attempt: balance, renewal, and sustainability. The partnership between humanity and microbes is ancient but entering a new phase of awareness. We no longer view them as enemies or curiosities, but as teachers. Their resilience, adaptability, and precision reveal the principles that sustain all living systems. The future, in many ways, will be microbial, not in domination but in harmony. If we listen to the lessons they offer, we may rediscover what life itself has always known, that progress is not a race against nature, but an alignment with its smallest and most enduring architects. From the first spark of metabolism in an ancient ocean to the living world that surrounds us today, the story of microbes is the story of continuity. No other form of life has persisted so long, adapted so broadly, or influenced so much. Their lineage stretches back more than 3.5 billion years, unbroken by ice ages, extinctions, or catastrophes. Through every transformation the planet has endured, microbes have endured with it, adjusting, rebuilding, and passing the thread of life forward. Continuity begins with replication, the simple act of one cell dividing into two. That pattern, repeated countless times across time and space, forms the heartbeat of biology. Every living thing inherits not only genetic material but also the microbial legacy that sustains it. The first bacteria to capture sunlight set the template for all photosynthesis. The first to metabolize oxygen paved the way for animals to breathe. Even now, the mitochondria inside every human cell carry remnants of bacterial ancestry, a living reminder that evolution is not a series of replacements but a chain of cooperation. In this light, extinction itself becomes relative. Individual species rise and fall, but microbial lineages rarely vanish entirely. Their genes persist, copied and reshaped in new forms. The photosynthetic machinery of cyanobacteria echoes in the chloroplasts of plants. The enzymes of ancient archaea power the metabolism of modern life. DNA, transferred and repurposed, acts as memory, a molecular archive recording every successful experiment evolution has ever made. The continuity of microbes also safeguards the continuity of ecosystems. When forests burn or oceans change, microbial communities are the first to recover. Their resilience stabilizes the environment, allowing larger species to return. They are the planet's immune system, restoring balance after every disruption. In a sense, microbes are time's repair mechanism, ensuring that even when the surface of the Earth seems broken, the processes of life continue beneath it. This persistence has philosophical weight. It suggests that life is not a series of separate events, but a single unfolding process. Each cell alive today belongs to the same ancient lineage that began when chemistry became biology. The diversity we see, from whales to moss, from fungi to coral, is variation within continuity, the expression of one underlying principle, the tendency of matter to organize, persist, and renew. Humanity, too, is part of that chain. Our bodies host ancient microbial partners, and our technologies increasingly rely on them. The oxygen we breathe, the soil we till, the food we eat, and the medicines we use all depend on microbial work. To recognize this connection is to see ourselves not as isolated observers but as participants in the same ongoing process. Life does not belong to us, we belong to life. Scientists now trace microbial ancestry across geological strata and meteorite fragments, finding echoes of familiar molecules even in the oldest rocks. Some speculate that similar life could have arisen elsewhere under comparable conditions. If that is true, the continuity of life may extend beyond Earth, a cosmic thread linking worlds through the chemistry of replication and adaptation. In every direction, the pattern remains the same. Life persisting through transformation. Death, decay, and destruction are not interruptions but transitions, feeding the same cycles that began billions of years ago. The microbes that consume a fallen leaf, that digest food in your body, that drift in the upper atmosphere, all play their part in this endless renewal. Continuity, in its truest sense, is quiet. It does not announce itself with creation or catastrophe. It flows like breath, constant and unnoticed, sustaining everything that exists. The smallest forms of life have carried that breath through ages of darkness and light, ensuring that, whatever happens, the rhythm of existence continues, steady, silent, and eternal. Every story of life begins and ends with microbes. They were the architects of the first ecosystems and remain the caretakers of the planet today. From the oxygen in the air to the fertility of the soil and the stability of the climate, their influence surrounds us, invisible yet complete. The human body, the forests, the oceans, and even the clouds owe their existence to their steady labor. When we speak of life on Earth, we are, in truth, speaking of the microbial world that made it possible. The legacy of microbes is written not in monuments or fossils but in continuity, the flow of energy and matter that has never stopped. Their ancient chemistry set in motion the cycles of carbon, nitrogen, and oxygen that continue to sustain every living thing. Even after mass extinctions, microbes endured, rebuilding food chains and restoring balance. When the first plants colonized land, microbes prepared the soil. When animals evolved lungs and digestion, they followed as symbionts and guides. They were not observers of evolution, but its engineers. Modern science has begun to see this clearly. Genomic research reveals that the DNA of all complex life carries microbial ancestry. The enzymes that drive photosynthesis, respiration, and replication are microbial inventions, preserved through time. The mitochondria that power every heartbeat were once free-living bacteria. The chloroplasts that turn sunlight into sugar are descendants of cyanobacteria. Even our immune systems, our senses of smell and taste, and our capacity to digest food reflect ancient collaborations with microbial partners. The human story, viewed under this light, is not separate from the microbial story. It is one of its chapters. Their legacy is also a lesson in humility. Microbes do not conquer, they adapt. They do not seek control, they maintain balance. In their patience lies wisdom. While civilizations rise and fall, their chemistry remains unchanged, governing the same reactions that began before continents had names. They teach that endurance does not come from power, but from flexibility, that survival is a matter of rhythm, of adjusting, waiting, and continuing. As the world now faces environmental change, the microbial perspective offers hope. If life can persist through ice ages, asteroid impacts, and shifting oceans, it can adapt again, with our help or without it. The question is whether humanity will choose to live in alignment with the systems that microbes perfected: recycling, cooperation, and renewal. To follow their example is to understand that balance is not stillness, but movement guided by restraint. The invisible web of microbes continues to surround you even now, in the air you inhale, in the water you drink, on the skin that connects you to the world. They move, divide, and rebuild, creating the conditions for your life in every moment. Each breath you take completes a cycle that began billions of years ago, a quiet exchange between your body and the microbial realm that sustains it. As you rest now, you may notice that breathing patterns settle into calm, each inhale and exhale a reflection of those same exchanges: oxygen and carbon, giving and receiving, balance and renewal. The microbes of the earth have been performing this rhythm since the dawn of time, and they will continue long after the night has passed. In their stillness, there is persistence. In their silence, there is creation. The legacy of the microbial world is not distant history but living presence, the quiet foundation upon which every leaf, every breath, and every heartbeat depends. In their patience lies the continuity of life itself. We, from Sleepy Stories, wish you a good night.